The overall goal of this experiment is to characterize yolk sac hematopoietic progenitors and their progeny in vivo using a mouse tamoxifen-induced fate mapping system in flow cytometry.
This method can help answer key questions in the immunology field concerning the development and differentiation of hematopoietic progenitors that are distinct from hematopoietic stem cells. The main advantage of this technique is that the hematopoietic progenitors labeled within a limited time window and their progeny, such as resident macrophages, can be monitored during development and adulthood.
Generally, individuals new to this method will struggle because of the low yield of cells per embryonic tissue and the large volume of the simultaneously treated samples.
To begin the experiment, first transfer dissected mouse uterine horns into a 10 mm Petri dish filled with approximately 30 milliliters of ice-cold PBS. Immediately after, grasp the uterus muscle layers at the cervical end of the horn and slide fine scissors between the muscle layer and a decidual tissue to release the embryos. Then, with fine forceps, remove Reichert's membrane and remove the placenta. Gently dissect the yolk sac and transfer it to a 24-well tissue culture plate filled with 0.5 milliliters.